Hello everyone, I'm Ting Wen-Chen from Institute of Bioinformatics and System Biology in National Yangming Jiao Tong University. First of all, I want to thanks for the organizer to give me this opportunity to share some of our works with you. And today I'll talk about identification of cancer prognostic biosignatures from multiomics data. And first things first, as you may already know that the DNA molecule is where the place cells store the genetic information. And all the genetic information are stored in the form as nucleotide sequences, as shown here. And the genetic sequences is actually quite simple, actually surprisingly simple, and is made of only 4 letters, as A, T, C, G, 4 types of nucleotides, as shown here. And the order of these four types of nucleotide decide the phenotype of an organism. And the different organisms might have different numbers of nucleotides in their genome. For example, in human, we have 3 billion nucleotides in our genome. And even though there are some neutral genetic variants in the population, for example, people may have different looking, and people might share different polymorphisms in their genome. Some changes in the important position might cause disease. For example, the most famous one are the mutations found in the cancer, actually results in cancer. Okay, and now we already have a technology called next generation sequencing that can be used to decipher the order of the nucleotides. For example, here we can use next generation sequencing to know the order is TCAA. And now we know next generation sequencing can be used to decipher the order of the nucleotide. I want to talk a little bit about what is multiomic. The information in cells need to be transferred. So the genetic information in DNA can be transcribed into mRNA, which stands for messenger RNA, shown as red ribbon here. And the mRNA information stored in mRNA will then be translated into protein, shown here as different colored circle. And so we have DNA, which is genome, and mRNA, which is transcriptome, and then protein, which is proteome. This kind of omics data, genome, transcriptome, proteome, and other omic data, epigenome and metabolome data, the information that we may use to determine the difference between disease and the healthy state. So analyze this kind of different omics data are called multi-omics analysis, which is the main technology that can be used in current medical study. And because in our lab we mainly use NGS technology, so today I'll focus on genome and transcriptome data, how we deal with these 2 types of data. So in genome data, usually we are interested in identification of the variants. As I already mentioned that in a human genome we got 3 billion base pairs, and usually we got reads from the NGS data, which the length can be 150 base pair. So we will need to mapping our reads to the human genome, and then we will have something looks like this one. And then we will be interested in what's the difference between the organism and the reference. It's shown here on the bottom side is the reference genome sequence, and on the upper side is the reads we identified from our sample. So you can see that here there is a difference between the reference genome and our sample. In the reference genome, we got A here, and in our sample, we got G here. That means we have A to G changes in our sample. And whether this kind of change is neutral or this kind of change is harmful, you will need to get some more information from the genome. And for transcriptome data, usually we are interested in identification of those differentially expressed genes. And as an example shown here, say we have normal samples represent as blue circles here, and we have mutated neuron cells represent as red circles here. And say we only got 3 genes in our genome, show here a DNA, we got only 3 genes, and those DNA can be transcribed into mRNA, and we have the expression pattern in our normal cell looks like this one, and the transcription pattern in our mutated cell looks like this one. And then we will be very interested in the second gene, because this second gene doesn't express in normal cell, but expressed a lot in the mutated neuron cells. So identification of this kind of differentially expressed gene might help us to identify those genes that play key roles in the disease. So in real life, when we want to identify those variants in our genome, we need to do all those steps in our lab. And using a toolkit called the Genome Analysis Toolkit, GATK pipeline. And using this kind of GATK pipeline, we can map our reads to the genome and then identify those highly confident regions of variant and annotate those regions, annotate whether those variants are important or not. And the result will look like this one. And each row here is one potentially cancer-related gene. This example is from an oral cancer example. And each row here represents one gene that might be important in oral cancer development. So, for example, the first one is EP.16, which is a very famous tumor suppressor gene, and usually got mutated in the cancer cells. So we need to identify the position and the type of mutation on TP53. For example, here we got many different mutations in different positions. So after we identify those kind of mutation patterns in our sample, we can try to search. We can try to identify potential biomarkers from those data. As for transcriptome data, we also try to identify the differentially expressed gene. Usually we compare normal sample to our tumor sample. Normal sample here I show as blue bar here, and the tumor part is here. So each column here is one sample, and each row here is one differentially expressed gene. And from comparing our data, we identified 3,548 differentially expressed genes. And we cluster those genes, and using those genes to cluster our sample, we can clearly cluster our tumor sample away from our normal sample. And if we further analyze what kind of pathway those genes involved, we can find that those genes are involved in cell matrix interaction, interferon signaling pathway, and so on and so forth. And those pathways might be important in oral cancer development. So again, we will need to identify those important genes in those differentially expressed genes. And after we identify some important key genes in our analysis, we are interested in whether those key genes are important in the clinical outcome. So usually we will do many analyses, and survival analysis is one of them. In survival analysis, we usually plot this kind of KM plot. In the x-axis is the years, how many years the patient will survive, and in the y-axis is the percentage of patients still alive. So we usually divide our patients into two groups. The first group is the one having higher expression level or having higher score of our scoring system, and the blue one here is the one having lower expression level of the target gene we are interested. So as an example shown here, you can see that the red group are doing much better than the blue group. That means the current oral cancer treatment strategy actually doing quite well for the red group. But for the blue group, we need to develop a different novel treatment strategy for this blue group, because they are actually respond badly to current treatment strategy. So I'll skip the other data. And in summary, I talked about what we did in our lab. And the most important step is actually to map the reads to the genome and to calculate how much confidence we have for calling a variant or calling a differentially expressed gene. And as I mentioned, that the human genome is quite large. We got 3 billion base pairs in our genome. So usually we did this kind of job in our high-performance computer, because you need huge computing resource to accomplish this kind of analysis. And after we identify the mutations or those differentially expressed genes, we may try to identify biomarkers from those genome or in the transcriptome data. And those biomarkers can help us to stratify our patients. And after we stratify our patients, we can further give them different customized treatment. So this kind of step is actually the first step for precision medicine. If you want to achieve customized treatment or precision medicine, you need to identify these kind of biomarkers to stratify your patients. And the example of oral cancer I talked about is a work we done in 2013 and already published in Nature Communications in 2013. So if you are interested in that example, you can look up the paper. And I want to thank many people, including my previous colleagues and my cooperators in ND Anderson and BCM. And of course, I also want to thank my lab members, and I want to thank the Ministry of Science and Technology and the Ministry of Education for their support for my studies. And that's all. That's all for my presentation. Thank you.